Hello and welcome to the second episode of the Synwave podcast. Today, the hosts are me, George, the president of the team,
Vas, the secretary of the team.
and we're interviewing our principal investigator, Dr. Julien Reboud. So let's start. Dr. Julien, what's your role in the University of Glasgow?
Hello, it's great to be here. My role formally, I'm a professor of biosystems engineering at the university, actually in the School of Engineering. And my role very primarily is 50% teaching and 50% doing research.
Yeah, it seems like you're a very active individual in the research field. But first, we were wondering, we want you to tell us more about yourself. So, what's your background? How did you end up in the university?
We can do a very long history. So no, I mean I always wanted to work at the interface between biology and physical sciences, but mostly towards the biological end. And actually, I was considering when I was in high school doing medicine as a career, but my whole family was in the medical sector. So my dad was a cardiologist, and my mom was working also in that area. And so, actually, that sort of convinced me that the work-life balance that I could see in my family induced me not to go for medicine. And I ended up in an engineering school, which in France is a well-favored track for career building. And in the end, the engineering was very general. So I was not getting the biology that I really wanted to get, and so I sort of created a master's at the end of the degree in collaboration with a university in Paris. And so I did a master's in biophysics at the same time as the engineering final year, which is an MEng in France, similarly. And after this, I went for a master's project for the final part of the course in a lab in Grenoble in France, and they were doing biochips, and we were working on uh, making new ways of developing drugs on cells, on biological cells. And I really enjoyed this. And I asked my supervisor whether there was a chance of doing a PhD, and she said no. So fine. But I really liked the research angle, and so I actually tried to find a PhD, and I went to lots of interviews, and it was always not ending favorably. And by that time, my supervisor actually decided that I could do a PhD. And so she invited me back, and I did my PhD with her um, for the next 4 years. And in France, normally it's 3 years. And the reason it was 4 years here is because during the PhD, we thought that the technology that we were developing was quite interesting and useful. And so we wanted to create a startup company. And so I took charge of that. And we had a bit of financing for that. So I took leave from my PhD to work on the startup project, and that lasted for a little bit of time, but in the end died natural death. In startup terms, it's 1 into 10 or less that go through, and we were not that one. And so at the end, I finished my PhD. And when I finished my PhD, I didn't really ask myself what I wanted to do, because my head of institute said, "Oh, I met these people from Singapore, and I thought that you would be perfect to work there. So I give them your CV. Is that okay?" And I said, "Sure." And I got an interview, and I went to Singapore. And so I did a postdoc in Singapore, more on so it was more microelectronics, and I was bringing the kind of biological angle to that. And we did a lot of work in diagnostics at that time with some companies, but mostly within the institute. But Singapore is, I mean, it's a fabulous place. It's really dynamic, and there's lots of energy, but it's also very tiring, and it's very far. And so my family was not too happy with us staying there for so long. And so at the end, we decided to come back. And so I applied to lots and lots and lots of different places. Actually, when I say I applied, My wife applied for me for most of them without telling me in the first instance because she really wanted to go back. And then we discussed, and we ended up going back. And so the first one as a postdoc I had was in Glasgow. That led to lots of work in acoustics, which is something that I had not done before. And it was really interesting. And we ended up creating a startup company on that as well during the postdoc, which I led. That also died. And we moved on, and so we ended up finding a new way to deliver drugs to the lungs using acoustics. And that was something that was really interesting. And actually, that also ended up as a startup company. And that startup company is still going on. So that's it's called Nebuflow, and it's not yet dead, in one way of putting it. And in terms of iGEM, what I did that linked me up to that competition is the fact that I was really interested in synthetic biology, because, as I said, I always wanted to work at the interface between engineering and biology, and synthetic biology, that is exactly what it is. And so I did a lot of work in artificial cells, so creating, using engineering systems, but putting biology inside these kind of droplets, and creating artificial cells in this way. And then I got working with another colleague, Dr. Sean Collins, in the life science department, and he got approached by some students from his side of the university to do an iGEM team, and that's the beginning of the story.
Yeah, I mean, that sounds amazing. It was a great background to get from Dr. Julien. I just kind of wanted to ask, are you able to talk about the current research that you have going on? Since you had mentioned that, your time split 50% teaching and 50% researching.
Yes, so as I said, I do a lot of work in diagnostics at the moment. So we develop new diagnostic systems, so medical diagnostics to diagnose disease, primarily in infectious disease. And actually, at the moment, we work a lot in Africa. So we develop point of care devices, so devices that are simple to use, low cost, and can give an answer relatively fast, within 1 hour. And so we are working with a lot of partners on a project called DDIDA. And that is with people in Africa, people in Netherlands, France, etc. And it's a really fantastic project with lots of people with lots of energy, to try and develop these devices. But the idea is always, I mean, doing the research is great, but it's about how can we move these into actual use. And so we're also working with some people in Kenya and industry, especially GADEX in the UK, to actually try and make that a reality. So that's one big part. There's also the work that I mentioned on delivery that I do with the startup company. So I'm still involved in that, and we try to do a little bit of work around that. And there's then significant amount of work in imaging. So we develop new imaging techniques to look at tissue, to look at cancer, for example, or at the moment we've got one which is quite interesting. A PhD student doing a system to take pictures of the throat and diagnose group A strep, which is a disease that gives you tonsillitis sometimes, or bacteria that gives you tonsillitis. And it's very difficult to diagnose, and so she's trying to do that and using artificial intelligence to try and make that easy. So you can see quite broad work, but mostly in diagnostics.
So, since you mentioned you work with diagnostics, we wanted to ask you a question regarding that specifically, since with our item we're also working with diagnostics. Yeah. We were wondering if you have an idea of what are some essential principles that make the perfect diagnostics. So, are there some characteristics that, when put together, they make the optimal diagnostic test?
That's a very interesting question. The first thing I will say, so optimal is a much better choice of words than perfect. Perfect never really happens. But in practice, our view of what is optimal or working well It's something that is accepted by stakeholders. And what does that mean? It doesn't mean that the technology is fabulous and can detect one molecule in a very complicated sample. It's a technology that is good enough that people will use it. And so that means that you've got to think more with the user in mind instead of thinking about the technology. And that's just a very personal idea, personal situation. There are lots of great research that's been done on, or is being done on, detecting or developing new techniques to detect molecules, cells, etc. That are really amazing, but they are a little bit behind in terms of the stakeholder in analysis and moving forward with that. But that's not, that's just not what I am trying to do. So it is about trying to fit something that eventually will make a difference. But the eventually for us, it's more towards maybe 2 to 5 years, where for some other techniques, I mean, let's say, quantum technologies are really fantastic, but they are not going to be for diagnostics on the market tomorrow. They'll be a little bit later. And so that's really what we think.
I just wanted to kind of backtrack a bit. Since you are our PI for the iGEM team, I think it would help our viewers kind of understand, what is your role as our PI, and what do you bring to our team, and like what leadership do you provide for us?
Yeah, that's an interesting one, because I mean, we've done a few iGEM teams, especially with Sean, over the years, and the role, I mean, we work very well as a team, so it was great. But the role of the PI changes depending on the team, and that's always really fascinating to have a look at and to live through as the iGEM team goes. In some cases, the role of PI is dictatorship. You are there to make sure that things happen, and you have to make sure that people come in and that they do the experiments, that they think about their research, etc. That's one end of the spectrum, and the other end of the spectrum is more trying to coach people and students in the team to go in the right direction, but without interfering in terms of the scientific ideas or the way the team works, because that's iGEM is not only a competition, it's also a huge experience that builds skills in group work, skills in experimenting, skills in design of experiments that are you can't get in any other way very easily. And so we do have to take a step back on that. But in this particular context of more like this team this year, the way I see my role is more as a facilitator. So the idea that if there is a problem, I can identify it, when sometimes it takes a bit of effort, and we can solve it together. And if there is a need for a specific resolution that is not directly obvious, or that requires contacts with other scientists or other organization, then I just try and facilitate that.
Yeah, and I feel like you've been guiding our path, but also letting us think our own ideas of how we want to solve this problem, and that's good because it allows us to develop a connection with each other, to really work together as a team. And you mentioned that teamwork is also a big part of the iGEM competition. But on that, would you say that the most fun thing about the process of participating in such a competition is the teamwork, or is it something else?
The more fun, you said, right?
Yeah,
Is that the word?
yeah.
So that's a very tricky question, because it will depend on the team. But I think actually that the more fun that I've had in iGEM is when you go to iGEM, because it's true that it's great to be working as a team here in Glasgow, and the team will do very well, I'm sure. But there are some challenges, and so you have to manage that. it usually works very well, especially when the lab work starts. You get a lot of different interactions then, which are much more interesting and often more fun, because people tend to take liberties with that, which is great. But that just doesn't compare with getting into. I mean, I've never been to the Paris version of iGEM, but the Boston version was really fantastic, and you get so much from it that it's really amazing. The challenge of the team is the stress. So when you get towards the end, where you realize there's still quite a long way to go, the stress goes high, and that impacts on the team dynamics, which is often challenging to manage. But it's a learning process. It's very important to learn how to react to that. And so the fun kind of decreases at that point, right? But it decreases also when you go to the competition, because that's a competition, right? And so you're stressed. But after you've done your pitch and you've seen the judges, right, then that stress is gone, and now you are enjoying it, and it really is truly a great time.
Yeah, continuing on with your experience going to the Jamboree, are you able to like mention any standout projects that you saw at the Jamboree?
I mean, it's difficult. First, I don't remember most of them. No, I mean, the projects that we were doing in Glasgow were really interesting and really fun. The projects that won most often were really good and really interesting. I must say that I mean, that's just coming from my culture as an engineer. I'm less enthused by projects that are dry and that do a program to enable something. Although the impact of these could well be very high. It's just I don't get excited about it. But that's just my own personal feeling about these things. No, the project I remember. So the great thing about iGEM is that it's not just the science. I mean, I know everyone gets really excited by the science because it is usually quite interesting. But the great thing about iGEM is that it puts the science in the context of what it needs to actually create impact. And the one thing that they took very early on that other people were not very good at is the ethics and the stakeholder engagement. And so you have to talk to the stakeholders. You've got to, and you have to, if you want to actually qualify for anything. And so that kind of opens the eyes of the students to lots of things that they wouldn't have thought about being in their scientific learning situation. And I really enjoyed the one project that we did in Glasgow, which actually the best part of it had nothing to do with the science. The science was actually quite straightforward. But the best part was the community engagement, and the students made a comic book, they made a mascot, they made a prototype that was a plush toy that glows in the dark. The project was very complicated, but the kind of enthusiasm of the team for that was I mean, I've not forgotten about it, and it's something that I will remember. But it was really interesting.
Yeah, I really liked when you mentioned that within the competition we sort of step out of just the science field and we try to view science in the scope of a global scope almost through the community, and we're really stepping. Well, I can say it for myself, working in this team. Besides the science stuff, I've stepped out of my comfort zone, trying to network and communicate with so many people to organize all this. And that reminded me of your backstory growing up. As you said, I noticed that you mentioned a lot of resistance in doing what you wanted to do. So you started with engineering, whereas you wanted to do biology, and then with the PhD issues and the startups. So as most of the team, we're new to the university and we're finding our pathways. Is there any advice you would give to us, or maybe your younger self, in finding what you want to do and how you want to do it?
Yeah, it is a difficult question to answer because it's always very hard to go back and say, oh, I should have done it this way or that way, or you should do it this way because actually no one knows. It's going to evolve. It's life. It just stumbles. But what going back um, onto the career choices that I've made, if you, I mean, it's a podcast, so you can re-listen to what I said. But basically, I didn't choose anything. I just went ahead, right? And whenever a decision came, I just said, I mean, I didn't always say yes, but I said, that's interesting, right? And let's go and try. And actually, that's an attitude that I stand by, and it served me, and it has worked so far. Um, but it may not work for everyone. That's difficult. But what I would say is that if one puts too much pressure on choices, they end up never being the right one. Whereas if you just think about it reasonably, and then there is no direct um, wrong turn that you can take, then just take the one, and then it will be the right turn. But if you think back, oh, I should have done this one instead, right, that's very dangerous, and it's not really the case because you don't know what would have happened, and it's much better to just go through and make the most of where you are at that point. But it's about this attitude of trying and making the most of what you want to do.
Yeah, so if you see an opportunity or a door, you should just keep looking forward. And even if it's something that you're not used to or you've not done before, there's no loss in trying. It might be the one, as you said. Yeah, I like this. I like this. I have an interesting topic. So I was talking to my friends and saying, "Oh, I'm interviewing Dr. Julian. He's a professor in my university. Are there any questions you would like to ask him, just to see what people outside of the field would like to ask?" And one of my friends, he's really into computer science. He sent me an article about AlphaFold. If you heard of it, it's an artificial intelligence model that predicts the structure of proteins. Um, so, moving into that direction, do you have any opinions on AI and how it can be implemented and used by scientists in a synergetic manner? Do you think that's possible?
Yeah, I mean, it's AI is making huge strides at the moment in any field really, but in biology in particular, it's working very well. Um, that that is linked to the ease of implementing it in some ways, which has kind of performed a step change. And this particular program that you mentioned, AlphaFold, the beauty of it is that it does work. And that wasn't a given at the beginning, and it's was lots of hard work. But it means that you can indeed take a sequence, so the amino acids that form the protein, and just put that into the program, and it gives you the shape. And the shape of a protein is directly linked to the function. And so that's interesting because you can sort of go from DNA directly into a shape. But that's less useful, right? Because you can just predict something, and sure, but what does that give us? The real interesting thing about AlphaFold is the opposite way. You have a function, so you sort of know the shape. How can I get back to the way of making it, which is DNA? And that's a challenge, but it's very promising. And so one example of where a similar type of AI is being used extremely efficiently is to combat antimicrobial resistance. So we have lived with antibiotics my whole life and your whole life, but it wasn't before, and it's now looking like it will not be in the next few years because we are running out of options. The bacteria and the pathogens are evolving out of the sensibility to antibiotics, and the antibiotics stop working. So we need to create new ones, but the pharmaceutical industry is having real trouble with that. It's very hard to create new ones that would work, and also it takes very long to actually make them safe to use and to release them in practice in the clinic. And so there is a lot of work in trying to get that process done much faster and to create new classes of antimicrobials. So molecules that don't work in the way that the previous one worked, because we know that pathogens will evolve out of that. And AI is being used to do that, because you can tell, or I can use this particular function maybe to create an antimicrobial because it's not been done before. There's no background for the AI to do, but actually it starts to think about everything that is being looked at in databases, etc. And they say, oh, you should try this. And so far, there's been a few papers, especially on peptides, so antimicrobial peptides. The papers are very promising, and they are easy to make, and apparently some of them have not been, they've not found a resistance pathway or mechanism to them. So it's something that actually could be built out of resistance. I mean, it's truly exciting AI. It's really, but as with every tool, although it is exciting, you've got to use it in a particular way, and you've got to be careful, because very often for these systems, so AlphaFold in particular, it's something that predicts, right? So you've got to check how the prediction happens, because if it happens just completely by random, then the result that you get is maybe the right one, but that's just chance, right? So you've got to really understand it. So we call that explainable models, and so there's a lot of work around that as well.
Yeah, and there are limitations to it, of course. After our research, we found that yes, it can predict the structure of the protein really accurately, but it cannot predict how that structure might change after the protein interacts or after its function. So we still need the knowledge we have as scientists to use this tool. It's a tool. It's not something else. So we have to think about how we use it. Yeah, that's good. I think it's quiz time. Is it quiz time?
Yeah, sure. Let's do quiz time.
It is quiz time.
I've been dreading this.
We know you're excited. So it's time to find out if you're on par with the GCSE biology students. We found this quiz by BBC, and it's a quick fire quiz of 10 questions about biology, and we will see if we can answer all of them. Are you ready?
Not really, no, but we'll make a start.
Let's do
We'll
this.
get started then.
So read the question for the audience.
Which of these processes do not occur as a result of aerobic respiration in animal cells? Generating movement, producing ethanol, building larger molecules, maintaining body temperature. Oh, that's very interesting. So this is the second one, producing ethanol.
Let's see.
But you see, these quizzes are very tough because the question is does not. You know, it's very, they're crafted.
As our teachers used to say, you should always read the question well. So it's correct. Great. Let's move on.
What is relatively large in a single-celled organism? The surface area to volume ratio, the nucleus, the volume, the mass. In a single-celled organism, what is relatively large is the nucleus.
Let's see. Oh, that's incorrect.
The nucleus is very large. So, you know, in a normal cell, the nucleus is basically the biggest thing inside the cell. Yeah. So I'm disagreeing with that final answer.
You're basically right.
It's true that but it's true that the surface area to volume ratio is big. So maybe it's the word organism that is different, but it's interesting. What subcellular structure must water diffuse across during osmosis? A gradient, the nucleus, the skin, membrane. I will say membrane.
I think that should be right. Yeah.
That's correct, yeah. But that wasn't.
Don't say that. What is not used in organic farming? Oh, that's a GCSE question.
Yeah,
that's very interesting.
I didn't know this was taught in GCSE.
Crop rotation, free-range animals, manure, pesticides. Well, I mean, it's a sort of general knowledge question. That would be pesticides.
Yeah.
Let's see. That's
Nice.
correct.
Very good.
So your general knowledge is good as well.
What form of contraception stops the maturation and release of eggs for months or years? The contraceptive pill, the implant, diaphragm, or condom? So, stop the maturation and release of eggs for months of years. I think they want to say implant because the pill is days, etc., etc. Let's see. Yeah, very good.
Great.
What is excess glucose stored as in the body? Starch, oils, glucagon, or glycogen? And that's glycogen.
Maybe you could get into Glasgow Uni if you took the exam now.
What is a dummy drug given in the drug trial called? Oh, that's a strange way of calling this, but that's okay. So, a painkiller, blind, antibody, or placebo, and I will say placebo.
Placebo.
It's also a test of my English pronunciation, by the way.
It is. Yep.
How many parts are present in a binomial name? I've never heard about this, but I'm just going with it's binomial, so it's bi, so it has two.
Who could have guessed? It's correct. Yeah.
Which letters are used to denote female and male sex chromosomes? So there's a range of answers with Y and X and different combinations. And so we are going to go for female XX, male XY.
No, I clicked the wrong one, but you're
clicked the
correct.
wrong one.
Yeah.
And the final question is, what gas leaves the blood in the lungs? Is that nitrogen, helium, carbon dioxide, or oxygen? And the gas that leaves the blood in the lungs is carbon dioxide.
Correct. 8 out of 10, but it's 9 out of 10 because I sabotaged you. So you have you're on par with the GCSE students. Someone could say. Could you say that?
Yeah, I guess. Well, we'd hope that you're on par with the GCSE students.
Do you know what we're going to do? Is I'm going to give that test to the first years
and see how much they get. Yeah. Hopefully, they also are on par with the GCSE student.
But yeah, you could get into our university if you were sitting our age. That's great. That's great. Now to move on to some more interesting questions. We have some questions about the team to see how well you know us.
Oh my God.
Or how well you've been following our path up to now. So let's start with, we believe, a relatively easy one. Do you know when was our first meeting as a team?
How do I know that? That's very tough. I mean, it was in the first semester, so it will be in 2024. I mean, I will say, I don't know, October.
Close.
Quite close, yeah. It was actually November, but you have to keep in mind our team started back in 2023 because it was first year, first semester. I think the next question would be, do you remember who won our Christmas card giveaway?
I don't remember. I will tell you why I don't remember. Because it was a very messy situation. So this was at the end of the weekly meeting, and there was some food, I think.
He made his famous brownies.
Yes, and there were card games, and no one could agree on the rules. So although someone won, I'm not entirely sure we are clear that they won.
Even though it's an excuse, we will take that. We will take that because we organized it. So, and then final question. That should be a really easy one. So maybe we'll have another one after that, but we'll find out. What's the degree that studied most? Wait, I'll rephrase that. What's the degree that studied by the most of our team members?
That is biomedical engineering.
Yeah,
yeah, that was a really easy one. Should we ask the last one or?
Yeah, so we had to double check with our treasurer, but do you remember who our treasurer is? And he said that he's willing to give you a clue
No, don't give it yet.
Not yet, but
I have no idea.
he said, "Do you remember a Belgian man?" It was Alex. He said one of the striking conversations that you guys had was that he told you that he was from Belgium, and you said that, yeah, I can see that, and that he looks Belgium. do you have any comments to add to that?
Now, there is a lesson in this, right? Which is that you should always be careful of what you say when there are other people around.
And that concludes quiz time. So we're very happy to have you here, and maybe you'll be in one of our future episodes as well, since you're a principal investigator. Is there anything you want to say to our viewers to close the episode?
Yeah, no, I mean, I enjoyed myself. It was very interesting to see how little I know. But I really want to say that the team is going very strong this year and has lots of motivation. I mean, for one, people show up, which is fabulous and really well noted. But they show up not only just to show up, but they've done the work and they're proud of the work that they've done. And I think you should be proud of leading a team like that, because it is going very well. There are lots of challenges ahead, and there will be lots of stress. And as I said at the beginning, some of the team dynamics will change, and there might be some difficulties along the way, but I'm sure we'll make that, and we'll get to Paris, and it will be great.
Thank you. This has been George.
And this has been Vas.
And we're going to see you on the next episode of the Synwave podcast.